Hi, welcome back. So in this chapter, we will cover the neuroscientific study of development and aging, such as the major structures of the brain and their main functions, and major theories of age-related changes in the brain. In ancient history, the brain was not considered very important. For instance, the ancient Egyptians thought that the heart was the source of human wisdom and the center of emotions and memory. With the advancement of neuroscience and neuroanatomy, we now have an understanding of the brain in more detail, and that each hemisphere of the brain has at least 180 distinct functional areas. We also have discovered that the brain can change over time with age. Our brain triples in size during the first year after birth, and by the time we are age 2, our brain reaches 80% of full size and weighs approximately 3 pounds by the time we become an adult of 25 years of age. Our cerebral cortex, which is responsible for higher level thinking, accounts for 85% of the total weight, and over 75% of that weight is water. The major structures of the brain are depicting the cerebral cortex, the corpus callosum, the prefrontal and frontal cortex, the cerebellum, hippocampus, limbic system, and the amygdala. Our brain contains about 100 billion brain cells called neurons, and this is about the same number of cells as there are stars in the Milky Way galaxy. Neuroanatomy is the branch of anatomy that specifically deals with the structure of the brain and the organization of the nervous system, whereas neuroscience is the study of the brain. It's a more broad interdisciplinary field that explores the nervous system structure, function, development, and pathology. It encompasses various subfields that investigate how the nervous system works at different levels, from the molecular level to the behavioral level. There are two neuroimaging techniques that are often used in studying neuroanatomy and neuroscience. One is structural neuroimaging, which provides highly detailed images of an anatomical feature of the brain. Structural neuroimaging includes X-rays, CT scans, and MRIs, and they document what a specific brain structure looks like at a specific point in time. Structural neuroimaging is usually effective at identifying bone fractures, tumors, and other conditions that cause structural damage in the brain. Functional neuroimaging provides an indication of brain activity, and so this includes PET scans, fMRIs, and NIRSIs. fMRI is the most commonly used technique, and it provides researchers with information about what parts of the brain are active in real time when people are performing specific tasks. A typical image will show different levels of brain activity as different colors. So for example, red on an image might indicate high levels of brain activity in that region, whereas blue might indicate low levels of activity. Researchers take three main neuroscience perspectives in studying the neuroscience of aging. One is the neuropsychological approach, and this compares healthy older adults with those with pathological disorders of the brain. So this provides information about whether damage in specific brain areas elicit cognitive deficits that are comparable to healthy older adults. The goal is for researchers to find out whether a decline in cognitive functioning has to do with underlying changes in the same specific areas of the brain that we observe in people who have pathologies or disorders. So this allows researchers to determine whether cognitive decline is part of the typical aging trajectory of healthy older adults. The neurocorrelational approach links measures of cognitive performance to measures of brain structure or functioning. Some researchers investigate correlations between medical assessments that are associated with the function of specific brain regions with behavioral outcomes. Because this is correlational, we can't ascertain whether assessment tests of brain functioning accurately reflect the actual function or anatomical activity of specific brain regions. So typically this is where we don't have enough tests, or we have a limitation of tests. And the only thing we can do is link certain measures of a person's performance or behavior to measures of brain activity or structure. The third is activation imaging approach, which links functional brain activity with cognitive behavioral data. So this is more of a real-time approach by looking at changes in brain function while a person is performing a particular task. For example, younger adult brains tend to show activation in one hemisphere of the brain when they are performing specific tasks, but older adults' brains tend to have higher activation in both brain hemispheres when performing the same tasks. The brain is very complex and is made up of various components. So we will learn briefly how the brain is organized. But at the most basic level, the brain is made up of neurons, which are cells that contain key structural features. These structural features are dendrites, axons, neurofibers, and terminal branches. Dendrites act like antennas to receive signals from nearby neurons. The axon is a part of the neuron that contains neurofibers, and neurofibers carry information inside the neuron from the dendrites to the terminal branches. The terminal branches are the endpoints of a neuron. And so when information gets released and passed on from one neuron to another, the terminal branches release chemicals called neurotransmitters. Neurotransmitters travel across the space between neurons, called the synapse, where they are received by dendrites of the next neuron. And so in order for neurons to fire and receive information, they have to travel across the synapse. And because neurons do not physically touch each other, this is how information gets passed from one neuron to another. Each region of the brain has distinguishing features that relate to the specific functions that those regions control. The prefrontal and frontal cortex is involved in higher order executive functions, such as the ability to make and carry out plans, switch between tasks, and maintain attention and focus. The cerebellum is located in the back of the brain, and it controls equilibrium and coordination of fine motor movements. The hippocampus is located in the middle of the brain, and it is a key structure associated with memory. The limbic system is a set of brain structures involved with emotion, motivation, and long-term memory. For adult development and aging research, the most important components of the limbic system include the amygdala and the hippocampus. Some age-related changes occur in neurons. So as a person gets older, the number of neurons declines. The number and size of dendrites decreases. Tangles typically develop in axon fibers. There are increases in deposits of proteins. The number of synapses decreases, and these changes occur in greater numbers in diseases such as Alzheimer's disease. An important structural change relating to neurons is the declining health of the brain's white matter. This is termed white matter hyperintensities. White matter refers to neurons that are covered by myelin, an insulating sheath that transmits information from one part of the cerebral cortex to another, or from the cerebral cortex to other parts of the brain. White matter hyperintensities are determined by the observation of high signal intensity or a bright spotty appearance on MRI images, which indicate brain pathologies. White matter does show some normative deterioration with increasing age. A neuroimaging method known as a diffusion tensor imaging is a type of magnetic resonance imaging that assesses the rate and direction that water diffuses through the white matter. This results in an index of the structural health of the white matter. Dopamine is a neurotransmitter that's associated with higher level cognitive processes. These would be inhibiting thoughts, attention, planning, emotion, movement, pleasure, and pain. And collectively, these neurons that use dopamine are known as the dopaminergic system. High dopamine levels are linked to cognitive processing that is effortful and deliberate. And so, because dopamine is associated with these higher level cognitive functioning processes, declines are related to poor episodic memory and tasks that require higher level cognitive functioning, such as attention planning and inhibiting thoughts. And so with age, this has shown that dopamine has been associated with these declines. Declines in the dopaminergic system are also related to acquiring tasks that must be performed quickly and the amount of information that can be held in mind at any given moment. There are cognitive tasks that require cognitive effort, which we will go over in Chapter 6. And in contrast, fewer age differences are observed in more automatic tasks, such as juggling familiarity with information. Overall, the studies using neuroscience methods to examine changes with increasing age suggest that changes in dopamine levels play a major role in cognitive aging. There are other neurotransmitters that we will go over, such as serotonin, which is involved in several other types of brain processes, including memory, mood, appetite, and sleep, and those are also related to age-related changes. Abnormal processing of serotonin has been shown to be related to cognitive decline. And so the neurotransmitter serotonin is involved in memory, mood, appetite, and sleep, like I stated in the other slide. And so this is related to cognitive decline, both in normative aging and in dementia. Others are disorders are sleep problems and schizophrenia, which are also related to a lack of serotonin. And so increasing the functional level of serotonin does have beneficial effects for certain problems. There is also another transmitter that plays a role in aging, and this is acetylcholine. This plays a role in general cognitive processes, such as arousal, sensory perception, sustaining attention, and higher order thinking. So damages to structures that use acetylcholine are associated with memory declines. They are also associated with the development of cardiovascular disease. So acetylcholine does have an important role in not only cognitive processes that play a role in dementia, but also in cardiovascular functioning that's controlled by the autonomic nervous system. One important change that happens during older adulthood is that the brain shrinks by late life. And it generally shrinks about 5% per decade, starting at around age 40, and then increasing after age 50 in most people. The cerebral cortex will get thinner over this same time period, and so you have considerable shrinkage which occurs in the brain. However, there are some changes that are not uniform for people. The most important arena in this complex pattern is the interface between emotion and memory. And so one intriguing research finding is that brain pathways that are involved in memory tend to deteriorate with age, whereas pathways involved with emotion do not. So for example, the positivity effect is being motivated to derive emotional meaning from life and maintaining positive feelings. This seems to increase during older adulthood. So older adults tend to show more motivation to derive emotional meaning from information compared to younger adults. However, theory of mind is more the ability to understand that others have different beliefs, desires, and viewpoints from our own. And so although older adults are more likely than younger adults to attend to emotional meaning of things, their ability to understand others or have a sense of empathy tends to decline. And so theory of mind is more about having emotional intelligence and characteristics that help us understand another person's perspective and viewpoints. So research studies indicate that these abilities increase during childhood but show some age-related decline in adults over the age of 75. There's also evidence that executive functioning tends to decline in older adults. Older adults have more difficulty focusing on relevant information, and so they tend to focus more on information that's irrelevant and have a hard time ignoring information or background noise. And this is likely due to reduced volume of the prefrontal cortex. There's also memory changes, so specific structural changes such as the hippocampus can result in memory decline. However, adults have better emotion regulation as they get older. So many areas of the brain are intimately involved in the creation, storage, and retrieval of memories. And among the most important of these are the frontal, temporal, and parietal lobes, the hippocampus, amygdala, and the cerebellum. And so there's a lot of research that's examining links between memory and specific structural changes in the brain. So as individuals age, a lot of these structural changes tend to take place and impact how memory performance and executive functioning. As individuals age, the medial temporal lobe shrinks normatively, but this shrinkage is markedly increased in certain diseases, such as those with Alzheimer's disease, and this helps explain why severe memory loss is a hallmark of this disorder. So there is an increased processing of positive emotional information with age. And this could be due to the fact that with older adults, they have more brain activity between their prefrontal cortex and their medial temporal lobe compared to younger adults. So these increases in connections may be due to age-related changes that occur in the prefrontal cortex, which make it necessary for older adults to use more connections and process the information. There's also social emotional cognition. So we have specialized areas in our brain known as the orbital frontal cortex, the amygdala, and basal ganglia, and this allows us to process social cognitive information. So this allows us to make social judgments, and it's more of a relatively automatic system in which we're able to read cues in the environment quickly and easily without deliberation, and then we make social judgments. So for example, if somebody is staggering down a hallway, we may automatically assume that the person may be intoxicated without taking into consideration that there may be other factors that might cause somebody to stagger. And so we have a tendency to automatically put the person into a pre-existing sociocultural category. So we make judgments that are easily activated and well practiced. So older adults may rely more on automatic judgment processes than reflective processing. So researchers have presented some evidence that drawing quick conclusions in ambiguous situations is a result of how our brains are wired. And so as we get older, we tend to process more social cognitive information and make more automatic social judgments. So humans are hardwired to quick process social cognitive information in particular ways that reflect well-rehearsed prior learning. So again, with age, these brain structures involved in more automatic processing have less age-related deterioration, whereas those involved in more reflective processing have more severe deterioration, which is why older adults tend to make more automatic judgment processes. Interestingly, research has also shown that the older brain may be compensating for this deterioration in cognitive decline. So for example, there's some studies that show that when presented with similar tasks, younger adults exhibit focal unilateral activity in the left prefrontal region, And older adults exhibit bilateral activity, which is both left and right prefrontal areas. And so the question is the older brain working harder and recruiting more brain structures? Or is activation merely the ineffective attempt to inhibit irrelevant information that results in the interference of optimal functioning? So it seems to be that older adults are compensating for this decline, and supportive evidence comes from the association between the additional activation in older adults and higher performance, which is not found in younger adults across performing several different tasks. So there is greater prefrontal bilateral activity in older adults during working memory tasks compared to younger adults. And so you'll see in the next figure, there is a typical left lateralized prefrontal activation pattern that's observed in younger adults. And on the bottom left side is the typical pattern found in older adults, and they are engaging both the left and the right prefrontal areas. So from this figure, it appears that frontal bilateral activity increases with age, as seen with the frontal bilateral activity in older adults during a verbal working memory task on the left, and then in older adults with higher performance in a long-term memory task on the right. Some authors have reported that older adults are more efficient than younger adults in processing on certain picture naming tasks because they have developed a more efficient strategy, relying on a more experienced, specialized, and segregated naming pathway. However, more frontal lobe activity was not seen in all situations, so researchers concluded that task factors may lead to the development of more effective and efficient processing. And in this case, older adults appear to apply the repeated experience of picture naming to reconfigure their brains to operate better. And so contrary to stereotypes, older adults' brains do have an ability to improve. There are several theories and models of brain behavior changes across adulthood. There's three most prominent theories. One is the parietal frontal integration theory, one is the crunch model, and the stack R model. And these models make common assumptions. And the assumption is that the primary reason for greater activation in different brain regions is the need for the recruitment and involvement of additional brain structures in order to successfully execute cognitive function as a person gets older. So the parietal frontal integration theory, it proposes that intelligence comes from a distributed and integrated network of neurons in the parietal and frontal areas of the brain. And so older adults sometimes exhibit reduced activation or recruitment of appropriate prefrontal regions, and they can sometimes exhibit the same or more recruitment compared to younger adults, depending on the task they are doing at the time. Based on the large number of studies that are using various types of neuroimaging techniques, the parietal frontal integration theory proposes that there are individual differences in intelligence, such as having origins in individual differences in brain structure and function. So, for example, a PFIT predicts a type of intelligence that's termed fluid intelligence, and it includes skills such as spatial and matrix reasoning. The PFIT model is an example of theories based on neuroscience research and has been tested and supported in several studies. And so it's clear that performance on specific measures of intelligence is based on how efficiently we can process information and are likely related to connections in the prefrontal and parietal lobe. The second theory is known as the crunch model, which is compensation-related utilization of neural circuits hypothesis. And this describes how the aging brain adapts to neurological decline by recruiting additional neural circuits to perform tasks adequately. And this incorporates the fact that there are additional mechanisms at work of the aging brain that's overutilizing other regions in the left hemisphere on demanding tasks. And so the brain has activation happening in both the left and the right hemispheres. And brain functions that cause behavior are controlled in both. So this is referred to as bilaterality of activation. So older adults are compensating for age-related changes by recruiting brain power from other hemisphere. And this model goes further by noting that bilaterality is not the only possible form of compensation. So for example, the amount of task-related brain activity directly parallels task demands until a person's cognitive resource limit is reached. The scaffolding theory of cognitive age was first proposed in 2009, and it is a well-known theory in cognitive aging research. And it proposes that as we age, our brains engage in scaffolding, which is compensating for cognitive decline so that we can maintain cognitive function in the face of neural challenges. And this involves recruiting additional neural circuits to support declining structures. And so the revised model is based on the idea that age-related changes in the ability to function reflect this lifelong process of compensating for decline. And so we compensate by recruiting additional brain areas and take life course factors that enhance or deplete neural resources into account. And so in the next visual, you will see a variety of life course factors that can either enhance or deplete our neural resources. And in chapter 6 and 7, we will go over this in more detail, but aging is associated with both decline and preservation of various cognitive abilities. And so what we know from neuroimaging studies is that during this lifespan, we are constantly scaffolding and compensating for these cognitive decline in order to meet cognitive demands or maintain cognitive goals. A default network of the brain refers to regions that are. Plasticity involves the interaction between the brain and the environment and is mostly used to describe the effects of experience on the structures and function of the neural system. So there is the belief that plasticity occurs in the brain, which means capacity of the brain is not fixed. It can be learned or improved with practice. So for example, people can learn ways to help themselves remember information, which can also help them deal with declining short-term memory ability with age. And so there are certain situations in which the brain compensates for age-related changes in processing by reconfiguring neural pathways and enlisting additional brain structures in processing. And so when you think about plasticity, it becomes a defining notion in development and aging that underlies the process by which people live in the world and they accumulate information and knowledge from experiences over time. And so those experiences in turn then improve cognitive functioning or sustain it. And so From a neuroplasticity perspective, research on stem cells has revealed compelling evidence that the brain is capable of generating new neurons and related cells when needed. Neural stem cells are cells in the brain that give rise to new neurons. They persist in adult brains and they can generate new cells throughout adulthood.